Hello everyone, we're going to get started today on the subject of mitosis. So jumping straight into it, mitosis is the process in which somatic cells or body cells use to reproduce or to duplicate themselves. So your somatic cells in your body will undergo the cell cycle, and after they've passed various checkpoints, they will reach a point where they are ready to divide. And that process of cell division is known as mitosis. So going straight into it, early prophase is the first form that you go into in mitosis. In early prophase, cell starts to break down some of its structures and build others up, setting up for the division of chromosomes. The chromosomes here, they start to condense, makes it easier for them to pull apart. And the mitotic spindle here starts to form between the centrosomes, which is the pair of centrioles. Now, the mitotic spindle is made of microtubules, which are basically strong fibers made of proteins that are part of the cell skeleton. Basically, the microtubules' job is to help organize the chromosomes and move them around during mitosis. And over here, the nucleolus, which you would have seen as a small kind of black dot within the cell, is disappeared. When this disappears, it means that the nucleus is also getting ready to break down. The nucleolus is the small inner membrane of the nucleus. The next phase is late prophase, or they call it prometaphase. In late prophase, the mitotic spindle begins to capture the chromosomes, and the chromosomes have now finished condensing. They're very, very compact. You can actually start to see them at this point. Also, the nuclear envelope breaks down and the chromosomes are released into the cytoplasm. And the mitotic spindle starts to grow more so that the microtubules start to capture chromosomes. Also, you'll see that the centrosomes have migrated to opposite points on the cell. Now, in metaphase, the spindles have captured all the chromosomes. They attach to the chromosomes and line them up in the middle of the cell, and the cell is getting ready to divide. These chromosomes align on this imaginary line called the metaphase plate, and at this stage all the chromosomes are lined up. They're getting ready to pull apart in opposite directions. And now it brings us to anaphase. In anaphase, the sister chromatids separate from each other and are starting to be pulled towards opposite ends of the cell. The protein that holds these sister chromatids together is kind of broken down and allows them to separate out from each other, and now each is their own chromosome. The chromosomes of each pair are pulled towards opposite ends of the cell, and microtubules that aren't attached to any of these chromosomes start to push against each other and elongate the cell. It makes the cell longer. It starts to stretch it out, kind of getting it ready for the next step. The next step, which is telophase. The cell is nearly finished dividing now, and it starts to, assume its normal structure. The nuclear envelope, the nuclear membrane starts to reform, the spindles start to disappear, and the chromosomes start to decondense again. You start to see the nucleolus reappear, and the cell is starting to very slightly pinch together, and it's starting to separate into two different cells. Which brings us to our last phase, cytokinesis, which is the division of the cytoplasm. This is the final stage of mitosis, and it can either start in anaphase or telophase, depends on the cell, but it does finish right after telophase. So you get this actin ring which starts to pinch the cell together and divide the cytoplasm as well as all the organelles into two separate cells. You end up with this cleavage furrow here. And by the end of cytokinesis, you should have a complete nuclear membrane again. Your chromosomes will be decondensed and you will have complete cells, two complete cells at the end of this. Thank you for watching. I hope you enjoyed the video.